So welcome everybody, welcome champions, FSHD Global Champions Group. This is June Kinoshita from the FSH Society, and today we have John Porter from the National Institute of Neurological Disorders and Stroke here as our guest. Thank you very much, John.
Sure.
And John will be giving a presentation titled Partnering for FSHD Therapeutics and Optimizing the Predictive Value of Preclinical Research. So without further ado, I will hand the podium over to Dr. Porter.
Okay, thanks, June. So I've got a lot that I'm going to go through today, but if people have questions as we go along, please jump in. We don't have to get through everything. I told June I'm happy to amplify on a subtopic in this in a subsequent call. But let's just, what I wanted to do is kind of give you an overview of a lot of territory here in thinking that we've had at NINDS about developing therapies and trying to help push things along, as well as the things that I've learned from partnering with advocacy organizations such as those groups that you guys represent on this call. So first I should say that as June said, I'm a program director at National Institute of Neurological Disorders and Stroke. This is one of 20-some institutes and centers that comprise the National Institutes of Health. Mm-hmm. So I don't do research here. I was actually in academic research for 21 years before coming here 8 years ago. What I do, I describe it mainly as helping facilitate funding in the fields that I cover for the Institute. So I cover the muscular dystrophies, spinal muscular atrophy, and myasthenia gravis, so a subset of the neuromuscular disorders, if you will. At times previously, I've also covered neuropathies, but with expansion in the Charcot-Marie-Tooth funding, that got to be a bit too much, so somebody else is covering that now. So I'm happy to talk with any of you at any time about ways we can partner together, because what I'm really trying to do is help your fields by helping investigators succeed in getting funding and then helping leverage each other's dollars to really make things happen for the field. Okay?
Yeah.
So, next slide, Gene. Okay, the problem that we all need to avoid. There was a paper a couple of years ago, Nature Review Drug Discovery, that even though phase 2 clinical trial success rates were already fairly low, they reported that phase 2 trials dropped to 18% success rate with insufficient efficacy, in other words, failure to meet the designed endpoint for the trial being the most common reason. Certainly trials fail because of toxicity and other reasons, but I think the thing to focus on here is that 18% success rate efficacy trials is really a pretty low success rate, and a lot of factors contribute to that. We're going to talk about a couple of them today, but I'm mainly going to focus on the idea that the next two papers, the next two bullets on this slide. So Bayer did a survey of their in-house drug development programs, specifically where they were evaluating therapeutic targets to see if they could progress with them and move toward a drug in the clinic. And a lot of their ideas came out of the published literature, of course. And what they found is when they looked at those preclinical animal efficacy studies and tried to replicate them internally, they could only replicate 25% of the published preclinical studies. It's an awfully low number. So what this says, that if you look at the first study that's published that says this particular drug has efficacy in this animal model, what the Bayer data says is that really better be replicated before you begin to move forward with further investments on that particular target. And Amgen reported very much the same sort of data not that long afterwards. I think Amgen's was mainly an oncology study, a review of oncology studies, but Bayer's program covered a wide variety of diseases. So the implication of these, when you look at the bullets, is are poor animal studies responsible for at least some of these clinical failures? I mean, certainly that's not the only reason. Maybe the animal model is not very predictive, and the study was really done very well, but it didn't predict success in the clinic. That is probably an unavoidable error, but I think the thing we want to try to avoid is the portion of those clinical trials that fail due to the animal studies being poorly performed. That's an avoidable failure, okay? And again, please jump in any time. I get rolling as I talk through slides, and just jump in any time if you have any questions. Next slide. Okay, so I took a very basic approach with some of these slides. What we need to understand is we're in the process, we as funders, and we have a lot of control in this as funders. The studies don't get done without them having our money. We can help mitigate any of these mistakes. And I put the definition of mistake in here because it actually is more malignant than you might think of that we made a mistake in this study. And if you look at this, errors caused by poor reasoning, carelessness, insufficient knowledge. This is exactly the types of things we're talking about in studies that are poorly done and thus potentially not predictive. So we want to avoid these mistakes because we're using them to direct very substantial future investments, both in the efforts in dollars of funding organizations and researchers, but there's also the ethical issues of subjecting patients to a drug or a biologic where, because of a study might have been poorly performed, we really don't have sufficient rationale to go forward and test that drug in the clinic and put subjects at risk. Okay? So this is all about making sure these sorts of things don't happen. Everybody okay so far? Any questions?
Yep.
Good. Okay. So next slide. So if we think about just what the critical factors for success in FSHD, I think you're really trying to work through this list. So first of all, the targets. And this means we have to know the key disease mechanisms. So I think, we've identified key molecular mechanisms in FSHD. There's been an integrated model of the disease published that's currently being tested by various groups. So maybe there's a reasonable start there. But if the target's wrong at the very beginning, you're basically not going to go anywhere, because if the target is not going to be disease mitigating if you modulate that target, say DUX4 levels, if it turns out that that's not really a key factor in the disease, it doesn't matter how much we modulate it, it's not going to be disease mitigating when we get it into patients. So that's kind of the first class. The second class, the two blue items are the two red items. I'm not colorblind. Are really tools that we need to have into place, and your organizations have been hugely responsible with helping people get these developed. So do we have mouse models? Do we have in vitro models? What are the quality of those models? Whether we're talking about allografts, whether we're talking about stem cells derived from patients, whether we're talking about a new animal model, those are really essentially drug discovery and development tools that we have to put in place. And of course, the other piece is that we've got identified candidate therapeutics that are developed through screening programs and optimized, if it's a drug, through medicinal chemistry, and then we've basically got something to work with to move along, and then you start getting into needing particular resources that academics labs may not have available, so that brings into partnering to a certain extent. The next two bullets are really looking at the clinical population. Do we have the ability to detect subjects with FSHD and stratify them for clinical trials? You know that a clinical trial might have to be focused very much on a particular subset of the population, that portion of the population that may show a response within the time frame of the clinical trial. So those factors have to be worked out. And I know that there's a number of efforts going on now with looking at natural history of the disease and in trying to develop care standards. I'm getting into the next bullet here, resources for the trials, that these things really have to be in place. And I'll make that point even more strongly on the next slide. But for now, I think you need to be thinking about everything from biobanks, registries, common data elements so that studies are comparable, care standards, because if the standard of care is not standardized, if you will, across patients, then that alone could influence whether and how much a particular subject responds to a drug or biologic in a clinical trial. So certainly those have to be put in place to help ensure that there's an even baseline for doing the trial and minimize variability. And then finally, outcome measures for the trial, and we'll talk more about those later. The big thing that I've really been pushing in my time at NINDS is that we're all in this together. No one has enough money to do everything that needs to be done to get a drug to market, and it's not going to happen without us really cooperating, and that we have the partnering occurring across all the stakeholders in the disease. Okay, next slide. Okay, so these are the things that really have to happen during the course of developing therapeutics. If we look at the middle box, I should say, that's the obvious one. Is we want to get to where we're efficiently and effectively developing drugs and biologics that are specific and disease mitigating for FSHD. Well, at the same time, the top box comes into play, that in the process of developing specific drugs, what we want to do is gain insights into the process of developing therapies for this disease, so that mistakes are shared. Anything that's learned about developing a specific drug, from whether a particular target is valid to what endpoint might be good for showing whether something works or doesn't work in the clinical population, that type of information gets shared. We're talking about like non-competitive type issues, not what the specific drug is and properties of the specific drug, but things that would carry over to testing any drug or biologic in FSHD. We really have to learn about that process. So there cannot be a clinical trial where we don't learn something from that trial that's shared across others trying to develop therapies in the disease. And then finally, the bottom box is that in the process of all this, we have to develop the infrastructure to do clinical trials, the expertise. And a lot of that only happens, some of it can be predicted. Like I said on the last slide, that we've got to worry about having endpoints in place. We've got to have registries so we know where the patients are and they can be accessed in adequate numbers so that you can convince industry to get into the disease. If you don't have these sorts of things in place, then it may be harder to get others interested in helping develop therapies through very expensive phase 3 trials. So a key point about that bottom box is what happened with myozyme development for Pompe's disease. Genzyme did a very good job at developing an enzyme replacement therapy. So this is the middle box. They efficiently and effectively developed a specific drug to treat the disease. But once they got it developed, I've talked with Ed Kay about this, and he's not with Genzyme anymore, but he was during the Pompe's program, is that they discovered that they really didn't know where the patients were, in which patient population they should use in their first clinical trial. And so it's something that Genzyme learned in the process, but what Ed says about it is that if they knew what a problem the lack of clinical trial infrastructure was going to be to development of myozyme, they might not have ever launched the myozyme program to begin with. So this is one of the few successful neuromuscular drugs thus far, and they might not have launched the program if they knew what a barrier this bottom box was. So, I think we have to all think about all three of these boxes simultaneously. That you tend to go to the focus on the developing the specific drugs and biologics, but we need to think about these other pieces. Okay, next slide. Okay, one of the problems here, and one of the lessons that we've got out of this, is that there's really very few novel drugs that have been developed to date, and I alluded to this in the last slide, for neuromuscular diseases. There's at least three that I'm aware of: Myozyme for Pompe's disease, Gamunex, which is for inflammatory neuropathies, and Riluzole for ALS. But that's about it. So when you think about all the different neuromuscular diseases, there's really only these three and maybe one or two more of diseases that have FDA-approved drugs, neuromuscular diseases that have FDA-approved drugs, despite a lot of effort going on. So that tells you that this is a rather severe hurdle, so that we really do need to collaborate and share experiences. We need to have a rigorous preclinical and clinical definitions for success to make sure that we're not blowing some up, distorting something at an early stage, saying that this is going to work without having adequate rationale, without having equipoise to test it thoroughly before we move things along. We need this repeatable developmental process more than just avoiding the same mistakes. I think we want to learn what works and what doesn't work and share that information. We've got to have good animal models. We've got to have good teams at the start, and then know what the end product is going to look like. That a lot of people refer to this as writing the label that's going to accompany the marketed drug at the very beginning of the program, so that you know exactly what you want to get to in a final drug. Okay, like I say, I talk pretty fast and I blow through things. Let me stop a second. Anybody have any questions so far? Everybody okay?
No, I'm fine. Fine.
Okay.
Yeah, that's good. Thanks.
Okay, June, next slide. Okay, so if we look at FSHD, then I think what's been gained about understanding a mechanistic target so far is really helping develop a good framework. Several years ago, we didn't really have anywhere to go, and now I think we certainly have hypotheses to test. So I think we can check the box that things are moving along pretty well. Not that this is done, but things are moving along pretty well. The second point is that we do have a lot of tools in development. I think each one of the things that I've mentioned here are all being under development. Maybe not fully widely available yet, but a lot of you have been supporting efforts for everything from mice to the allograft approaches, and there's a beginning of a trial readiness effort with FSHD. Certainly there's a good registry, and I know some other things are going on. I think our questions are the validity of the DUX4 target, and specifically, what's the therapeutic window of that target? So I know that Luke Kunkel's lab presented some data at the Wellstone meeting in Boston last year about DUX4 being developmentally regulated, and so the question is that if it's a really early target that has to be addressed even prenatally, is that addressable by a drug development program for FSHD? I mean, so we need to know that. We need to know the window through which we can manipulate the DUX4 target and have efficacy. If it's too early, then maybe something downstream from FSHD is targetable for therapy. But I put this as just when thinking about where the field was at, I saw this as a question.
John, Dan Krause, I just have one interjection.
Sure.
And firstly, I was a little late on the call, so I really appreciate your being here and all the support of NIH and NINDS for FSHD, and most appreciative of your efforts as well. I just wanted to point out that we sometimes talk about DUX4 as a target, and I just want to make a very clear distinction that DUX4 may be a part of a mechanism that gets a disease to where it needs to be. So that DUX4 is necessary for FSHD, and it may put FSHD where it's going to occur, but it's not necessarily the target. I think some people kind of think of this as it happened in the beginning and that's it, rather than it got the disease to where it needs to be, and then other things happen over time. But regardless of whether it's there or not, if we knock out DUX4, it will not mitigate the disease, instead of the aforementioned strategy. So I just wanted to sort of throw out that nuance, is that we often easily talk about DUX4 as a target, but I think in that clause, validation of DUX4 as a target should be, instead of validation of the disease mechanism. You understand what I'm saying?
Yeah, Dan, I'm, in making some of the specific points about FSHD, I'm just trying to be provocative a little bit here and get people thinking about some of the broader approaches. Really, my main goal with this talk is for people to think about some of the broader issues, like really making sure that we bring appropriate rigor to preclinical studies.
No, I agree totally.
So, but also to get people thinking about what's needed, like the next point, the trial readiness. And I put approvable endpoints, even though, there's much more to readiness, like I said, natural history, registries, biomarkers for trial readiness. But when you think about collaboration, so the myotonic dystrophy field is struggling with this approvable endpoints issue right now, too. I mean, they're a little bit further along in trying to develop a therapeutic, but they're going to have the same problem, I think, with that FSHD has, in that you often have a slowly progressive neuromuscular disease that getting to the right endpoint so that you can show a change in that endpoint in a clinical trial that's a year or less in duration is a really critical issue, and that's one reason why I'm encouraging folks to think about that very early. Like going back to my Pampas disease example, if Genzyme encountered a huge difficulty in having a measurable endpoint for Pampas, they might have dropped the Myozyme program. So I think it's incumbent upon the community, and I'm including myself in that, the NIH in that we make sure that we put some of these things in place so that when you do attract good biotech and pharma partners, that they see things to be encouraged about, because they know that they have to have these things in place in order to get a drug approved.
So just so you know, Ria Brogarden gave an excellent presentation in San Francisco on that whole story, and I think that it was very eye-opening to many individuals at the Champions meeting. I think Ria was pushing more the history, the chronology, and the financial aspects of it, and I think less so the need to organize, preemptively, that kind of thing. But just so you know, Ria gave about a 40-minute presentation that was just really astounding on the Genzyme, Pompe story.
Yeah, I was really sorry I missed that, and again, with my father-in-law, that was just a bad week.
Yeah.
But no, you and I talked when you were planning the meeting that having an example from another disease where they'd had some success stories would be good, and so I think that's a great example. The Myozyme example is a great example.
John, this is Howard. Are you familiar with what's going on in Huntington's disease? Because it seems, I'm not super familiar with it, but we donate every year because my mother-in-law had it. Yep. And it seems that they have put together a very well-organized collaborative effort that seems to be yielding results.
Yeah, are you referring to CHDI?
Yeah, that and also the Huntington's Disease Society. So that, there's sort of two competing organizations. But
yeah, so one of my colleagues just down the hall here, I mean, obviously that's an important disease for NINDS too, right? One of my colleagues down the hall is the program director for Huntington's, and I'm pretty familiar with. So I know Roby Blumenstein, who helps head up CHDI. He was on our council for a while, and yeah, so you know, they have a big help there in that that CHDI has a huge endowment. I mean, I think a huge endowment. They basically run a large program just off the income from the endowment. But yeah, I think I think what the point you're making, if I can use your comment to make my point, is that, you know, we're all in this together, and there's got to be crosstalk among different diseases. We have to, again, it goes back to this learning about the process of developing therapies. We've got to get better at that. NINDS is responsible for a large number of neurological disorders, number one. And number two, if you look at the rare diseases, what the FDA defines as rare diseases, it's something like 6,000 diseases, and only 200 and something of them have an approved therapeutic. So we are truly in this together, and we've got to make sure that we learn from what other diseases are doing. If I can use your comment to make that point, okay? Okay, next slide. Okay, so you know the challenges are really pretty obvious. Is that then if you think about this, if we're going to try to make things more predictive in terms of taking the animal. Like I say, you know, animal models are animal models. They don't have the disease. Some of them may not be predictive at all, but I think we have to make sure that the studies are done right, so that if we have a predictive model, it can help us identify what are the right candidates to take to the clinic. And so here's different reasons that they fail, that the animal model doesn't model the disease, the mechanism and the target from the animal doesn't modify the disease when you get to human subjects. Sometimes the properties of the drug, the pharmacokinetics and the bioavailability, they're species differences, and even though it might be absorbed well and hit the target, say in a dog, when the pharmacokinetics is tested, it might not translate well. The effect size might not be very robust, like you see a 10% improvement in some measure in the animal model. We have to ask ourselves, is that sufficient efficacy in the model to then move the drug forward? I don't have an automatic answer to that. There isn't an automatic answer to that. My point is we have to think about the endpoints and the magnitude and change in the endpoint that we should strive for in order to say, yes, I think we can move forward with this particular candidate. And then there's this failure to replicate, and this gets at the Bayer study that I had only on the second slide, that they could only replicate 25% of the published studies that they looked at. I think what we can change, it's very difficult to change the first four factors on this slide. The thing we can change is we can make sure that unintentional biases aren't introduced into study design. Now, the most critical example of that, next slide, is from amyotrophic lateral sclerosis, Lou Gehrig's disease. And I'll go through this really quickly, but you know, it makes a huge point. There's something like 50 publications that say drug X extends lifespan in this SOD1 mouse that's been used as a model for ALS. Now, many of those drugs then went very quickly to clinical trials, but nothing has shown efficacy in the clinic. So you could ask the question, is it the mouse's fault? Was the mouse not predictive? We have these 50 different publications that say all these different drugs, quote, work in the mouse, but then when we get in the clinic, all the clinical trials fail. So is the mouse a poor model? Well, I'm not so sure it's a poor model, and here's why. Well, we can't conclude it's a poor model for the following reasons. First of all, an independent organization, ALS Therapy Development Institute, TDI, financed heavily by the MDA, and maybe almost solely at this time by the MDA with Paul on the call here, attempted replication of published results on 70 different compounds and did this very objectively and very rigorously. They wanted to know why these failed in the clinic, so they were retesting them in the animal. None of those compounds could be independently replicated in ALS TDI's hands. So when they did the studies rigorously, they couldn't replicate the positive animal data that had been published. So I say, the mouse can't be blamed in this case. Maybe the mouse is a bad model, but you can't conclude that from these data because this strongly suggests the studies were done poorly. In fact, then there's that quote by the authors of that paper suggesting that most of what was published was noise in the time of survival of the mice that were tested. Okay, that's pretty severe indictment for that field. Next slide. So, I'd ask the question whether we can avoid or even tolerate such large failures in any type of muscular dystrophy. 30 failed clinical trials. I would say no, because what's going to happen is they obviously had to get companies interested in helping make investments to do some of these clinical trials, or certainly to let them test their drugs in ALS. And with that many failures, industry is going to conclude that there's something wrong with the models, the disease is refractory to therapies, and they're going to walk away from it. So you undertake all of these efforts to bring biotech, get them interested in your disease, and if poorly performed early stage studies then trigger clinical trials that fail one after the other, you're going to lose that industry interest. So that's really what the stakes are, from one of the types of stakes. I'd argue that there's several stakes in this. Obviously, you don't want to expose patients to a drug where the poor rationale that it might work. But there's this other piece, too, that you're going to lose the collaborators that you need. Next slide. So, I wanted to put a little levity into the talk here. So there's this quote from Paul Newman in Cool Hand Luke, and I'm not going to do the Southern accent. Well, no, it wasn't Paul Newman. Sorry, the captain wasn't Paul Newman. The captain was somebody else. I mistyped that, okay, in the movie. But he had this very strong Southern accent. And after Paul Newman was hauled back after trying to escape from the prison, he was beat up by the guards, and the captain said, "What we've got here is a failure to communicate." Well, that's really true here, that these failures to produce peer-reviewed data are in part a failure to communicate. So the peer reviewers have to have the information about how the study was performed. And us as grant reviewers have to have the information about how the study was performed, that it wasn't biased, and that you have appropriate data to go forward to the next tranche of funding or to go forward to a clinical trial. Okay? That's really what we're talking about here. And this is how we're phrasing this with NINDS, that it's a failure to communicate. Next slide. So, this is a study that was in Journal of American Medical Association several years ago, where they looked at greater than 500 preclinical efficacy studies in really top-notch journals. And they looked at whether the authors had done this list of things. So dose response, a high percentage of the studies, 70-some% it looks like, had done dose response. Measuring outcomes that were clinically relevant, yes, a high percentage of studies had done that. But look further down the list. So blinding, so that the experimenter doesn't know which animal got the drug or the placebo, around 20% of the studies. Only around 20% of the studies did that. Randomizing animals so it's not predetermined which treatment the animal gets. Important concept because you can bias. You can pick the healthier looking animals and give them drug versus pick the sicker looking animals and give them placebo, and then you get a result that's almost preordained, right? So randomization is really important for preclinical studies. These criteria are used all the time in clinical trials. What we're trying to do is push them to be used more so in preclinical studies. Okay? Let me stop again. Questions?
John, this is Bill Lewis. Are you going to talk about the Duchenne dog model?
Well, I'm not going to talk about it here, but we can come back, in a little bit.
The big question I had was, how has it been used? Is it practical? Has it been a benefit, or is it just an interesting sideline? Yeah,
Bill, can we hold that to the end? That one I'd like to come back to, okay?
Sure. Okay.
Because this point I'm going to lead right into. So the point is that it hasn't been communicated very well. Most of these papers, they might have blinded, they might have randomized, but they didn't say they did.
John, a question on this is: you say the preclinical niche? Do you bring that back even further, sort of mechanistic niche?
Yeah. So actually, yes, we're very interested in that. And actually, so since we put out our guidelines that I'm going to go into it in a second that you know about, since we put that out, a basic science journal, Nature Neuroscience, has adopted these guidelines for submissions to their journal. They have a checklist where the authors have to say whether they did certain things and where that's reported in the manuscript. Okay, and that's what we would love to happen. We're trying to push NIH-wide to get this incorporated to grant reviews, not just for efficacy studies, but as you suggest, for basic science. Okay, I know you and I talked about this in Boston, so I hope you're happy to hear that we're pushing for all NIH applications to be looked at. Okay, we'll see if it succeeds.
Can you post that Nature Neuroscience reference?
I can send it. Yeah, I can send it.
Okay.
Okay. Yeah, they actually had an editorial and then a checklist. So do me a favor and drop me an email just to remind me, because I have another call right after this one.
Sure.
And remind me, and I'll send that.
Okay. All right.
Okay, next slide. So reporting deficits are widespread, and that has an impact. Okay, this slide's a little bit more complicated. But so FK506, it's a drug that was studied in a lot of preclinical studies of stroke. Okay, they tested this very, very extensively as a potential stroke drug. So 29 preclinical studies of publications of the same drug is rather substantial. Okay. Now, so along the y-axis, what you have is efficacy. So did the study show more than 30-some%, that grayed out bar across horizontal? Did they show more than that level of efficacy in treating the stroke model? Versus on the y-axis are indicators of the quality of the study. The 0 through 7 indicate how many different quality measures a particular study reported in its methodology. Now, these quality measures are things like randomization, blinding, that the study's appropriately powered, did they report whether there was any conflict of interest or not by the authors, as well as some stroke-specific things like was the temperature of the animal controlled during the conduct of the study, those sorts of things. So what you see here is the larger the number of quality measures that the study reported, like blinding randomization, the less likely it was to show efficacy. So the studies that reported up to 7 measures of quality had almost zero impact in the stroke model. So what that says is control of bias does matter. You could say that the more biased the study was, way over there at the left, with 0 measures of quality reported in the publication, the more likely it was to show an effect, potentially bias. Okay? So we would strongly argue that paying attention and reporting these quality measures does have an impact. And if we push people to report them, we'll increase the overall quality in our grant applications to all of our organizations, in publications. We'll push them to pay more attention to doing studies in an unbiased fashion. Next slide. So that has these consequences. Resources wasted on non-rigorous studies, wrongly advanced or dropped targets. You might think if you were that one FK506 study, you took it into trial. If you were the other study that had the 7 measures in the group that had 7 measures of quality, you didn't take it to trial. Okay. The ethics of risking human subjects with candidate therapeutics where the rationale is poor or unevaluated. I would argue in some of these studies it's unevaluated because the study was biased. So the poorly justified trials also divert human subjects away from other potentially effective agents. And as I've mentioned before, pharma will abandon disease if there's too many therapies, or if it becomes clear to them that the disease isn't ready, or that the target's non-druggable. You know, they make those kinds of judgments fairly factually, and a lot of failed studies would convince them of that. Next slide.
John, can I interrupt? This is Kees for a moment.
Sure.
On one hand, you got me depressed about how do we ever get a proper series of clinical tests going for FSH.
Yep.
On the other hand, there must be some really strong lessons for us what we need to do now when it's not costing that much.
Yep.
To secure that when we go into clinical trials, we have a bigger chance of success.
Yeah, so that's, I mean, of course that's where we're headed. Yes, the picture looks dire, but I think what we're pushing for is the answer. So we had a workshop.
Okay.
We published this paper in Nature, and Story Landis, the Institute Director for NINDS, is lead author on this. There were 4 different program directors, me included, in organizing the workshop, and so we're authors on it. And we just had a variety of people from academics and advocacy and companies participate in this. And the conclusion was right there in the box at the bottom that if grant applications and publications should report on certain core parameters. Now, this is mom and apple pie stuff. This is really, really basic. Randomization, blinding, estimating sample size, doing a power analysis, and how the data was handled. So if you have missing data due to death of animals, that gets accounted for in clinical trials. There's the intent to treat condition in clinical trials. Let's bring that rigor that is already well established and working in clinical trials and bring it to preclinical studies. I think that's the answer. And so we emphasize better reporting of the animal studies as a way forward. But as I've already mentioned to you, we're looking to change, well, we have changed our criteria internally within NINDS, and we're looking to try to change the criteria NIH-wide. Paul can tell you on the call from MDA that MDA, at least part of MDA, has already changed some of these things for their grant applications. Do you want to say something about that, Paul?
Well, we've sent this paper and I guess the previous, the Sean Scott paper to all of our reviewers. We did that immediately after this paper came out in our last review cycle, and they're completely on board with looking at these parameters in the applications. When this came out, it was too late to send to the applicants, but now in this latest review round that we're in right now, we're getting the applications in now. I've sent every applicant who submitted a letter of intent this paper. And in fact, I put the link in my signature line. So basically everybody who gets email from me gets this paper and another set of guidelines that we give them. And we tell our applicants that they are going to be reviewed based on their considerations of this, I guess it's box 1 in this paper. So we're trying to get this cultural change through the community. What we haven't done yet is put an actual checklist box in our applications. I want to go one step at a time, but I think it's very possible we'll be heading that way.
And to review, to go back to Dan's earlier point, the part of MDA, the committee that Paul runs, has a lot of basic mechanistic applications in it. So indeed, and correct me, Paul, but MDA is applying these criteria even to very early stage stuff.
Yep.
Okay, next slide. So we actually put on the web our overall guidelines, and this is accessible. If you just Google NINDS Rigor Guidelines, a PDF will pop up with this list of things. And I'm not going to go through it, but it just to show you that there's a little bit more to it than just than blinding, randomization, and powering. But this we spent a lot of time with not wanting to be prescriptive, but saying, you know, things that really have shown to make a difference in therapy development programs. So it's not just the NINDS kind of passing the law down, but that there's, when you talk to someone in industry, they would agree with virtually every point, if not every point, on this slide. And like I say, these are, you can Google these, but also if June wants the link, I can send it to her and she can distribute it to everybody. Okay, next slide. So the path forward is this. Now there's the link for, okay, it's already in slides that, and June, you can distribute this set, which is enhanced a little bit from what I sent you previously. There's the link to our Rigor guidelines. We've educated our reviewers for our own study sections within NINDS. We had the workshop and then that publication. But my key point here is that one funder, even the NIH, you know, can push this. But what really is going to take is for all of us to get on board. That we really need to have better rationale and transparency to justify the human subjects trials. So for a lot of reasons, we need to do this better. We even admit that we can't solve this alone. So I think the solution is by all of us to work together. Mm-hmm. Now, the thing to feel good about here is there are two choke points. There are two places that will make things change. And that is funding, because the studies can't be done if somebody doesn't fund it, and publication, because no academic's going to do studies that they can't get published, because that's part of the survival culture, okay? So those two choke points are addressable. We had journal editors involved in our workshop trying to help them take on the idea of pushing for this in preclinical studies, and that's indeed how the Nature Neuroscience change came about, is that editor was involved with our workshop. But then as funders, like I say, we can really control this. The ideas that we're doing about making our reviewers look at it, Paul says he's making his reviewers look at it. I think it's a cultural change that is so obvious, like I describe it as mom and apple pie, that we need to do it. Okay, so the last slide is just a model for maybe how you do preclinical studies, from discovery study through an exploratory study, through what we call preclinical trials, where maybe before you go to clinic, you have an animal study that has fully clinical trial type standards to it, that you even have a predetermined primary outcome measure, and you say that the drug-treated mice must achieve X improvement in this particular parameter, or else this fails. Okay? So I'm not going to go through it, but that's just one way to think about it. Maybe you don't want to necessarily slap on the full rigor approach in a study where somebody wants to just say, let me see if this works. You know, let's just try this, okay? And that's always going to happen, but what you have to be careful about is you don't want to have those let's try this studies being the things that trigger the trials. You want to have the very carefully done study being the thing that triggered the trials. So I think this is an addressable problem in the disease, in our disease fields. And I think it's an approach that we can succeed at, and we're certainly trying to do this NIH-wide. So that's it. Jumping back to the dog question, the problem with Duchenne, like most of the rare diseases that I work with, the communities, is we don't have a drug succeeded in the clinic yet. So we don't yet know if measuring a certain parameter in the dog, in the dystrophic dog, and seeing that that improves by X%, we don't know yet whether that's predictive of success in the clinic. A lot of times, well, not a lot of times, the way that the animal models and what we use to say whether the drug works in the animal model, the validity of that is only going to be seen when you have a drug that's developed using those approaches works in the clinic. And that gives you retroactive validation of the animal model, okay? So the dog, you know, I think the dog is good for a lot of reasons. It's much more severe than the mouse in Duchenne. But it's a hard resource to breed and maintain and even do the efficacy measures. There's a handful of people that are good at doing those measures. But those dogs even have a pretty high perinatal mortality. And so I know that Joe Cornegie had some folks that worked for him for 20 years in helping with birth and survival of the pups of those dogs. So, you know, it's a tough thing. It's not going to be all over the place. And Bill, we don't yet know if it's going to be predictive because we don't have a drug yet that works in the clinic.
Yeah, well, my assumption has always been that the dog would likely be a better candidate for human models than mice and zebrafish and so on. But unless we've proven that the dog really does do the job, then maybe that's a false assumption.
Well, I think that's going to vary from disease to disease, though. You know, it's going to depend. I think you always make the best with what you have. So, like I say, the animal models are never going to be perfect. They don't have the disease, okay? Never. A mouse model of Huntington's doesn't have Huntington's, okay? But what we can fix is how those models are used, and that everyone understands what it means when you get certain data in the mouse, okay, or the dog, or the monkey model. You know, like people go to the dog often for pharmacokinetics because they can get better PK data in the dog than they can in the mouse just because of species differences. But your efficacy models may be only in the mouse or another model organism. I think we always have to use the tools that we have, and it's our responsibility, right, to try to help develop therapies for the patients, the populations that we interact with, and you do the best you can with the tools that you have. But that's precisely my point. I'd like to see us do the best we can with the tools we have.
I've got a question, John. Just sort of, Dan Perez. So first, I agree with everything you say, and I think it's everything that we need to do. But I look at different populations, like FSH, since the MD Care Act has $100 million put into it, and Duchenne probably close to $1 billion. One community is much more mature in terms of biomaterials and cell lines and resources, and the other one not. So there was a day when we had 5 LCL lines and 2 myoblast lines sitting in Coriell, and that was it. That was the total universe of biomaterials for FSHD, and so we've really worked hard to bring those materials, a greater set of materials, a better characterized set of cells, biopsies, whatever. So as you come in, so starting anew, diseases don't always have the same clarity in terms of the checklist.
Right.
And so I guess the question, I always look at it as a question of where are we going to get the money to build the foundations to lead to a better result, more rigor result. And so in terms of NIH investing, is there going to be a culture change as well in terms of, is you're going to require that somebody have 40 cell lines, that they also have the money to go out and get those?
No, we're not at that level. First of all, we haven't gotten into addressing in vitro studies yet. Okay. This is all in vivo. Mm-hmm. But what we will expect is that someone doesn't do a study with just 4 mice in the control group versus 4 mice in the experimental group and say, well, this is all I could afford, or it's hard to breed these mice, and so this is all we could get.
Well, we saw a lot of that in San Francisco on November 6th. I did this on 9 cell lines. I did this on 9 mice. I did this on 7 mice. The question I asked after every presentation, how many mice was, I got this great result, and well, how many mice? 6, 7, 9, 11.
Yeah, well, it comes down to a power calculation, right? So probably a lot of times they're not doing power calculations. So, the question to ask is so for a given endpoint, power analysis for a given endpoint, if you want to show a certain percentage change in that endpoint, based on the variability and everything, the math will tell you have to have a certain number of mice, and that's different, depending on characteristics of the mouse, on the characteristics of the endpoint, everything. But the point is, just did they do that? So the question we get that I've gotten from some other advocacy groups in other types of dystrophy was that, well, we have pilot funding programs and we can't afford to pay for this to be done fully rigorously. And then I say, well, then it's got to be treated as a pure discovery study, and someone better not jump to conclusions based on those data. It better be understood that it was an underpowered study, and in that it's going to have to be repeated with more rigor, essentially, with the appropriate standards. And the other aspects, the randomization and blinding, Dan, that isn't in. I was in academics 21 years, and that's not in the culture. And so I think what we have to do is try to push for change. And as I say, we as funders, plus the publishers, I think, are the ones that can make this happen. And fortunately, I think we are at a place where we can make things happen. In that Bayer paper, when Francis Collins, the director of overall NIH, testified to Congress, to the Senate Health Committee last year, I think last spring, he was asked by one of the senators about that paper. And isn't this a problem that so many of these studies can't be replicated? So this is like on everybody's radar now. There's been enough of those papers. People have tried to make these kinds of change before. I'm hoping that we can succeed in making the change now. If we can get NIH-wide to change its review culture, then I think that's going to be a huge step. But believe me, I think you guys getting on board makes a huge difference too. and we've talked to every group that will listen to us. I spoke to Health Research Alliance several months ago, and they were already tuned into this issue, but even some of the data that I took to them, that stroke study, for example, that the higher quality, the less likely the people were to show an effect, really has an impact. So I have to apologize. I had to schedule something right after this, so I'm going to have to jump to that other call. But honestly, I think some of you on the call know that I really like working with all the groups in the fields that I cover. If you guys would like me to be on another call to amplify on any particular subtopic of any of this, I'm happy to do that. Okay?
Oh, well, thank you so much, John. And
you
gave us a lot. You gave us an excellent launch pad for the Champions Working Group, which will start really tackling these issues amongst ourselves, starting, I think, with a call coming up in the next couple of weeks.
Good.
So that's wonderful preparatory. You just plowed the ground for us, and I'm sure we'll draw on your expertise and your insight again in future meetings. So I wanted to thank you. I'll be following up with some emails, and I'm going to stop the recording now.
Great. Well, thanks a lot.
Thank you. Please stand by.